Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is a virtual journal club type event that is also archived on YouTube. And we're privileged this week to welcome the David Lyden Lab. So as many of you know, David Lyden's lab has been contributing to extracellular vesicle research for a very long time and has made some important contributions. This is one of the more recent ones. And we're going to hear from Wang today. Haiying Zhang is also here, along with the PI, David Lyden. And so we thank all of them for joining today and for being willing to share this very exciting work. And as you know, extracellular vesicles are not alone in the world. They don't move through our body alone, and we don't always analyze them alone either. There are other particles too. And so we're going to hear a little bit today about EVs, but also about other extracellular particles that can contribute in disease situations. So Gong, thank you so much for joining. I just want to emphasize to everybody that you should put your comments and questions in the chat box. And we will get to your questions. We will allow you to unmute yourself, show your video if you'd like to, at the end of the presentation. Gong, thank you again, and please go ahead and share this exciting work with us.
Thank you, Ken, for inviting us to present our most recent work. Yes, I'm currently a postdoc at the Dr. David Lyden Lab, and working closely with Assistant Professor Haiying Zhang. And today, our presentation title is the Tumor-derived extracellular vesicles and particles dysregulate liver metabolism. Okay, so as we know, cancer is a systemic disease that can dysregulate the host homeostasis through metastasis or the release of soluble factors, including proteins, metabolites, hormones, and extracellular vesicles and particles, so short for EVPs, to local and distant organs. For example, cancer rarely metastasizes to the muscle, but the cancer-associated factors can still induce muscle atrophy and cachexia. Furthermore, cancer can also induce the anorexia, can induce the inflammation, pancreatic insufficiency, and also the thrombosis. Most recently, our work has identified that tumor-derived EVPs can dysregulate liver metabolism without liver metastasis. So EVPs are heterogeneous based on their size and also their biogenesis. Recently, our lab has identified the new particles that are secreted by the tumor cells, we termed as the exomeres. So there are also some other different kind of extracellular vesicles has identified previously, like the microvesicles, microsomes, exopheres, and also small and large exosomes. It has been widely studied how the tumor-derived exosomes can mediate the metastasis of to distant organs. So there are multiple cargoes have been identified in the extracellular vesicles, including proteins, RNA, and also the DNA. So most recently, we identified the palmitic acid, the lipid cargo in the packaged in the EVPs can be targeted to the liver in the context of the cancer to induce the liver metabolic dysregulation. So to study the systemic effects of cancer beyond the metastasis, we focused on two tumor models that preferentially metastasize to the lung, but not the liver, which are the melanoma B60F10 model and also the osteosarcoma K7M2 model. So first of all, we orthotopically injected these tumor cells into the mice and waiting for 3 or 4 weeks to collect the liver. The lung metastasis was confirmed by the H&E staining, and there was low liver metastasis being detected. Then we did transcriptomic analysis of these liver tissues and identified some common differently expressed genes, including 469 upregulated genes and also 315 downregulated genes. Using these common differently expressed genes for downstream gene set enrichment analysis, we were able to find multiple common differently regulated signaling pathways that occurs in the liver. So we could see that the inflammation, TNF-alpha signaling via alpha-kappa B were upregulated in the liver, and also the lipid metabolism associated pathways like the bile acid metabolism, fatty acid metabolism, and oxphos were all downregulated in the liver. So based on this RNA sequencing data, we were wondering if the liver metabolism was dysregulated in these tumor models without liver metastasis. To this purpose, we did metabolomic mass spec and also the lipidomic mass spec. So the lipidomic mass spec shown here, we can find the cholesterol, ceramide, and also the triglyceride. These are the hallmarks of the fatty liver disease that are all upregulated in the livers of the tumor-bearing mice. So we were wondering if the fatty liver was generated in the tumor-bearing mice without liver metastasis. Then we did body staining to visualize the lipid droplet in the livers of both models. So for example, we can see that the lipid droplet was dramatically accumulated in the livers from tumor-bearing mice, but not the control mice. So which means the fatty liver was generated in the tumor-bearing mice without liver metastasis. To validate the clinical relevance of these findings, we collected multiple liver biopsies from the pancreatic cancer at the diagnosis without any treatment. And we follow up this patient for up to 8 years, and we didn't see any liver metastasis, but only extrahepatic metastasis, such as the lung metastasis or the peritoneal metastasis, were identified after this follow-up period. And using these liver biopsies, we did RNA sequence analysis, and we could see that the inflammation signaling, like the inflammatory response, interleukin-6, JAK-STAT signaling, and TNF-alpha signaling were upregulated in the livers from the cancer patient. However, all the lipid metabolism such as pathways like the fatty acid metabolism and oxphos were all downregulated in a cancer patient, which is similar to the mouse models we have identified. And also, we did blood-based staining of these livers from both control and cancer patient. We can see that although they have similar or comparable of the BMI, but the body staining showed that the fatty liver was significantly generated in a cancer patient without liver metastasis compared with the controls. Since there were no liver metastases detected in both tumor models and the cancer patient, we were wondering if the tumor-derived extracellular vesicles and particles are responsible for the fatty liver generation. So on this purpose, we first isolated the EVPs from the tumor explant. So we cut the tumor into small cubes and cultured in the dishes for overnight, and we isolate the conditioner media for the EVP isolation. Then we labeled the EVPs with the fluorescent dye and intravenously injected these EVPs into mice. 24 hours later, we could see that the EVPs were enriched in the mouse livers. And to mimic the in vivo education of the tumor-bearing mice, we injected the EVPs into the mice via tail vein injection every other day for 4 weeks. Then these livers were isolated for transcriptomic analysis, metabolomic analysis, and also the lipidomic analysis. And we could see that the immune response associated pathways were upregulated, and also the lipid metabolism associated pathways were downregulated. Similar to the tumor-bearing mice, we were also able to identify the triglyceride, cholesterol, and the ceramide that are enriched in the EVP-educated mice. Furthermore, the body staining also confirmed that the fatty liver was generated in the EVP-educated mice compared with the controls. Then we want to know if EVP are essential for the tumor-induced fatty liver generation. So we knock out the EVP, the factor that responsible for EVP secretion from the tumor cells, the Rep27A, and in both the melanoma cell line and also the K7-2 cell line. And we could see that after Rep27A expression depletion, the particle numbers secreted from the tumor cells were dramatically decreased. And interestingly, we can see that the exomere was the largest population that suppressed after the Rep27A knockout, and also the small exosome to a less extent suppression after Rep27 knockout. And when we implant the tumor cells after Rep27 knockout, we could see that the EVP secretion suppression could lead to the suppressed the fatty liver generation in the tumor-bearing mice in both of the melanoma and the osteosarcoma models. Then we were wondering which particle or vesicle population in the EVPs are responsible for inducing the fatty liver. So we first out-centrifuge used this out-centrifugation to isolate the EVP first. Then we did the asymmetric field flow fractionation technology, which short for AF4 technology, and we were able to isolate exomeres, small exosomes, and also large exosomes population from the whole EVP population of the cells. And this is the EM picture of these different populations of the EVPs. Then we injected this EVP individually into the mice every other day for 4 weeks, and we could see that exomeres contribute to most of the fatty liver generation, and with less extent of the exosomes. So which means in the tumor-induced fatty liver generation, exomere contribute to the most of the functional changes. Then we want to know which cell population in the liver were responsible for the EVP uptake. We label the exosome with fluorescent dye, EVPs with fluorescent dye. Then we intravenously injected these EVPs into the mice. 24 hours later, the liver of the injected mice were subjected to flow cytometry. And we could see that there are almost 40% of the cells are positive for EVP uptake in both B16F10 or K7-2 EVP injected mice. Interestingly, most of the cell population that are responsible for uptaking EVPs are CD45 positive immune cells and also CD31 positive endothelial cells. And within the immune cells, there are more than 90% of those immune cells are Kupffer cells, which are the resident macrophages in the liver. And this finding was also confirmed by the immunofluorescence staining with the F480 and also the EVP staining. EVP is in red color and F480 standing for Kupffer cells in green color. Then we want to know if the Kupffer cells are essential for tumor-induced fatty liver generation. So to this purpose, we first orthotopically injected tumor cells into the mice, and after certain days, when the establishment of the primary cells occurred, then we intravenously injected the liposome or claudinate into the mice to block the Kupffer cells in vivo. So we could see that after the claudinate injection, most of the Kupffer cells were depleted from the liver. which were confirmed by the flow cytometry. And also we did body-based staining, and we were able to see that cloudinate depletion of the Kupffer cells rescued the fatty liver generation in tumor-bearing mice. This is the statistical analysis of the body-based staining. Kupffer cells, as the liver-resident macrophages, will be able to secrete multiple chemokines and cytokines after activation. So we first isolate Kupffer cells from the naive mice and validate its functional integrity by F480 staining of the Kupffer cells, and also by activating these Kupffer cells using LPS. After the time course, we could see the interleukin-6 was secreted from the Kupffer cells after LPS activation. So after confirming this technology of the Kupffer cell isolation, we isolate Kupffer cells from both the tumor-bearing mice and also the EBP-educated mice. And we were able to see that the TFR, the multiple cytokines and chemokines were secreted from the Kupffer cells in tumor-bearing mice and also after EBP education. TNF-alpha is one of the cytokines has highest secretion level compared with all the other cytokines and chemokines after the EBP education. And the TNF-alpha secretion was also confirmed in the plasma of the both tumor-bearing mice and the EBP-educated mice. So which means when we do the ELISA analysis of TNF-alpha on the plasma, we could see that TNF-alpha concentration was highly enriched in the plasma from both tumor-bearing mice and also the EBP-educated mice. Then we want to know how the TNF-alpha can contribute to the fatty liver generation. So on this purpose, we treat the primary hepatocyte with recombinant mouse TNF-alpha in vivo. So we could see that after TNF-alpha treatment in vitro, we can see the lipid droplet were accumulated in the primary hepatocyte. Mechanistically, this TNF-alpha treated hepatocyte was subjected to RNA sequence analysis. And we did the gene set enrichment analysis, and we found that the lipid catabolism, such as the fatty acid beta oxidation, lipid catabolic process, and also the fatty acid catabolic process, these lipid catabolism associated pathways were all suppressed in the TNF-alpha treated primary hepatocyte. Then we want to know if we block the TNF-alpha, can we rescue the fatty liver generation in the tumor-bearing model and also EBP-educated mice. So we first inject the tumor-bearing mice, we inject the tumors into the mice, or we educate the mice with the tumor cell-derived EVPs. At a certain time, when we establish the primary tumor, we inject the anti-TNF-alpha antibody in vivo multiple times every other day. At the end point, we collect the livers for body-based staining, and we could see that after TNF-alpha blocked, we were able to rescue the fatty liver generation in both tumor-bearing mice and also the EBP-educated mice. So what kind of cargo in the EBP were responsible for inducing TNF-alpha secretion from the Kupffer cells? We then did a metabolomic mass spec on the EBPs from the tumor explant. And we could see that multiple saturated fatty acid, including stearic acid and also palmitic acid, were significantly enriched in the EBPs from the tumor explant. And also we did free fatty acid mass spec on this EBPs from the skin explant and also the B16 melanoma tumor explant. We could see that the palmitic acid represent the highest enriched lipid saturated fatty acid that enriched in a tumor explant. Importantly, this finding was also confirmed in both the B16F10 cell line-derived EVPs and also K72 cell line-derived EVPs compared with their controls. So which means the palmitic acid is the highest enriched saturated fatty acid in the EVPs from the tumor cell. Then we treat the primary Cooper cells with the palmitic acid in vitro, and we could see that palmitic acid alone can induce the TNF-alpha expression from the Cooper cells. And furthermore, we want to know if we block the palmitic acid package in the EVPs, can we rescue the TNF-alpha expression in the Cooper cells? Then we treat the B16F10 cells or the K72 cells with the C75, the inhibitor of the saturated fatty acid synthase. So after this C75 treatment, the free fatty acid content in the EVPs were dramatically decreased in both of the cell lines. Then we use this C75 treated cells derived EVPs and also DMSO treated cells derived EVPs to treat the primary Cooper cells. And we could see that the less package of the palmitic acid in the EVPs cannot induce the TNF-alpha expression from the Cooper cells anymore. So which means the saturated fatty acid, especially the palmitic acid, is essential for the Cooper cell secretion of the TNF-alpha secretion from the Cooper cells. Then we want to know how the palmitic acid can induce the TNF-alpha expression from the Cooper cells. So we did screening of multiple receptors, and finally we found that the toll-like receptor 4, which short for TLR4, is responsible for the palmitic acid-induced TNF-alpha expression from Cooper cells. Actually, it has been widely reported that TLR4 are the responsible receptor for palmitic acid that can induce the downstream inflammation in multiple immune cells. And also we treated the primary Cooper cells with the TAK before the palmitic acid treatment. The TAK is an inhibitor of the TLR4. So when we inhibit the TLR4 signaling, we could see that the palmitic acid cannot induce the TNF-alpha secretion from Cooper cells anymore. Importantly, the EVPs from both tumor cells were not able to induce the TNF-alpha secretion after the TLR4 blocked by the TAK inhibitor. So we can conclude that the tumor EVP induced the TNF-alpha secretion from the Cooper cells depend on the functional integrity of the TLR4. Then we also did the gene set enrichment analysis, and it was interesting that we found the drug metabolism associated pathways, cytochrome P450 genes, were downregulated in all the TNF-alpha treated primary hepatocyte and the B16F10 melanoma tumor mice livers, and also in the livers from the cancer patient. And actually, we have known that the cancer patient, especially in late stage, they were too weak to stand with the standard dose of the chemotherapy. So which means they might not be able to metabolize the chemo, that they have very poor prognosis and also life quality. Interestingly, we did qPCR analysis of the multiple cytochrome P450 genes in the primary hepatocyte after TNF-alpha treatment, and we could see these CYP genes were all downregulated. And also these CYP genes were downregulated in both the tumor mice and also the EVP-educated mice. If we block the TNF-alpha signaling in the tumor mice and EVP-educated mice, the CYP genes expression were regulated. So which means the TNF-alpha can suppress the CYP genes in both the tumor-bearing mice and also the EVP-educated mice. Then we were wondering if the drug metabolism in the tumor-bearing mice or in the EVP-educated mice can be suppressed by the can be suppressed. So to this purpose, we sectioned the liver from the mice and to culture the live liver slices ex vivo. And then we treat the liver slices with multiple substrate of the CYPs, and the metabolite were analyzed by the mass spec to confirm the drug metabolizing activity. So with this technology, we could see that in the liver from the tumor-bearing mice and also from the EVP-educated mice, the drug metabolizing activity was suppressed in these livers. Importantly, when we treat these mice with TNF-alpha antibody, the drug metabolizing activity could be rescued. Since the drug metabolizing activity was suppressed by the tumor EVPs, we were wondering if the chemotherapy toxicity could be increased in this EVP-treated mice. So on this purpose, we first inject the mice with tumor cell-derived EVPs to induce the fatty liver first. Then we did the IP injection of different chemo. So which means we did decarboxylation treatment, which has been widely used for treating melanoma. And also we did the doxorubicin treatment, which has been widely used for treating osteosarcoma. So we can see here that compared with the control, the EVP education increased the chemotoxicity of these drugs, as shown by the bone marrow suppression, like the significantly decreased red blood cells, reticulocyte, and also the hemoglobins. Interestingly, for the cardiotoxicity of the doxorubicin, we can see that compared with the PBS control or the osteoblast EVP education, the K7-2 EVP education significantly suppressed the ejection fraction and also the fractional shortening of the cardiac function. So which means both the EVP indeed increased the chemotoxicity in the tumor in the educated mice. So in summary, we have identified that the tumor-derived EVPs that containing saturated fatty acid, specifically the palmitic acid, can target the Kupffer cells in the liver and to trigger the TNF-alpha expression and secretion from the Kupffer cells. The TNF-alpha in the liver can induce the inflammation microenvironment in the liver, which can induce the lipid droplet accumulation to promote fatty liver generation, and also suppress the cytochrome P450 gene expression to suppress the drug metabolism, which can increase the chemotoxicity. So this is very interesting that our finding our study finds that the tumor without liver metastasis, but with lung metastasis, can promote the fatty liver generation and suppress the drug metabolism. Actually, another interesting finding in our lab is that when the tumor has liver metastatic tropism, but not the lung metastatic tropism, the tumor-derived EVPs can also induce the thrombosis in the lung. So this is kind of highlighting of the systemic effect of the EVPs in the tumor beyond the metastasis. So at the end, I would like to thank Haiying and David for their support of this work, and also thank our collaborators and also all the lab members for their help with this project. Thank you very much.
And thank you, Gang, for this
Yeah.
presentation. This is really useful. David, do you have a few words to say?
Yeah, I just want to follow up. So when you have tumors that are promoting lung metastases, the majority of the exosomes are going to the lung and setting up shop for a pre-metastatic lung. And as Gang highlighted, it's the exomeres that always go to the liver. And we're still trying to figure out, like, how do they go to the liver and why are they uptaken by Kupffer cells? But as he highlighted, they generate the fatty liver. And so in models like pancreatic cancer, the exomeres and exosomes are both going to the liver, but the pre-metastatic liver always outweighs the fatty liver in those models. And so we have the patients that develop liver mets several years after a diagnosis of pancreatic cancer that we get the liver biopsies. They do not have fatty liver, but they have other problems supporting a pre-metastatic niche, such as vascular leakiness, and inflammation, like including net formation, which is neutrophil extracellular traps, and ECM changes. I also think, Gang, he didn't have time, but what's very exciting, if you have a fatty liver, there's secondary systemic effects from that fatty liver. You have increased triglycerides and cholesterol. So that's explaining why there's such a high cardiovascular disease in cancer patients. It's not just related to treatment. And so they could die of a heart attack as opposed to the cancer burden. There's low arginine, which makes T cells not function. So your immune system can be affected with fatty liver. And finally, as a third example, you have low creatine and creatinine, which the liver shuttles to the muscle. And so you could have cachexia indirectly with loss of muscle in your heart and diaphragm, which also can enhance mortality. So I just wanted to highlight those points that are explaining how cancer is really a systemic disease. And like for us to target, say, I'm just making it up, melanoma that likes to metastasize to the lung, you have to think, oh, I have to block maybe the exosomes or just, preventing lung mets, but you also have to target the liver and prevent fatty liver disease and all these secondary complications. So it seems complex, but this is what's happening in the cancer patient. We're not recognizing that it's a multi-organ involved disease, and we have to treat the future metastatic organ and these other organs that are also involved in systemic disease. So that's just my overall comments.
Well, those are not just overall comments, but a masterly, I think, painting of all of these relationships between the different parts of the body and how we have to consider those, and we have to consider how those different compartments are communicating with each other. So thanks for sharing that. Lots of good considerations there, David.
I have
We
a
have
question.
yes, we'll get to you in just a minute.
Sorry.
We're going to go through the comments in the chat box in order here. So I have now allowed unmuting, but please wait until I call on you, and we will get to your questions in order here. So we want to start with Yongkang Yang. Yongkang, are you there? Do you want to ask your question, please?
Very good work. Hi, Gang.
Yes.
I have a question about the technical injection. So for these EVs, how did you inject them into the animals every other days?
We did the tail injection.
You did type of injection. So how to guarantee all the EVs go to the liver? It should go to everywhere.
Yes, correct. So, but majority of the EVs will go to the liver when we do the biodistribution analysis.
Ah,
They go to multiple organs to function differently. Like we are also doing another project. Like they go to the lung, and they can promote the pre-metastatic niche formation in the lung to induce lung metastasis. So here we focus on the liver, and there is no liver metastasis detected, and we did see the fatty liver generated after the EVP injection.
all right. Thanks so much.
Yeah.
Thanks for the question. Next, Graciela, please go ahead.
Yes, hi, Graciela Glicum. I tried incubating cells with TNF-alpha, but I incubated them overnight and I didn't wind up getting the results I thought. So I'm wondering, how long did you incubate the cells with TNF-alpha?
I didn't do them overnight. I did them only like 4 hours in incubation, and the short time incubation I did the RNA sequence analysis. So we got the findings with short time incubation.
Thank you. That's very helpful for me.
Yeah.
Thank you.
Great. Let's move next to Pasquale D'Acunzo.
Hello, can you hear me?
Yes, we can. Please go ahead.
Okay. Yeah, you partially already answered, but first of all, very interesting work. And I was just wondering if this effect was liver specific. I mean, we know that it is going also in other organs. So I was wondering if you saw this accumulation of lipid droplets also in other organs and that contain, for instance, macrophage-like cells. And if not, well, what's your opinion why this does not happen, considering that, you know, also other cells can take them up?
Actually, this is really excellent question. Like if we can observe the lipid condition in multiple organs. So currently we haven't done that yet, but probably maybe, I mean, I expected there might be some if there's enough macrophages accumulated in the muscle. Since muscle during the cachexia development, there are also some fat deposition in the muscle fibers. So I was wondering if it's possible that this fat accumulation in the muscle could also be mediated by the EVPs from the tumor. So we have to do more like detailed study of the other organs.
I mean, I just want to add a comment that exomeres, for the most part, are going to the liver, and to a small degree, they're going to bone marrow and spleen. So if this is an exomere phenomenon, we wouldn't see it in other organs where they don't go. So like Yang's going to tease apart the other exosomes that are going to other macrophages, as you suggested. But I would predict it's going to remain mainly in the liver, maybe the spleen and bone marrow as well.
Thank you very much.
Can you skip my chat box talk? This is Phil Eskenays.
Hi, Phil. No, we're getting to you. We're getting to you. We got one, I think one or two ahead of you. So we'll get there though. No worries. We have the next question here from Roger Carlos Fontana, and this is, I really like this question because it has to do with the evolutionary considerations of the tumor.
Hi, yeah, good afternoon. Can you hear me?
Yes, we can.
Great. Yeah, first of all, thanks again for this great and very clear presentation. And yeah, so my question is a bit more about the biological reason behind what you're observing. So I was wondering if you could hypothesize on why do you think that these tumor cells produce these particles, EVs or exomeres that are rich in fatty acid, especially because like if it is like to get rid of them, like when people originally thought that EVs might be just like a waste disposal system. I'm not sure if it makes a lot of sense because palmitic acid has been found, I think, to enhance cancer cell proliferation and and metastasis. But then if it's not a waste disposal system, if it's not random, do you think it is purposefully aimed at inducing this liver damage that you observe? And if so, why or how do you think this might benefit the tumor? Just maybe a bit of hypothesizing about why do we observe this phenomenon at a biological level?
Yeah, this is interesting question. Thank you. So actually, in my opinion, at least from the data we observed compared with the normal cells, we didn't see much enrichment of the fatty acid in the EVPs compared with the tumor cells. For the tumor cells itself, to support its own highly proliferation rate and like the growth rate, it need multiple lipid fatty acid for its like membrane component. So during this process, the tumor cell itself can synthesize more fatty acid compared with the normal cells to support their proliferation. So this is why they might be able to package more EVP, more fatty acid, like the palmitic acid, into the EVPs.
Well,
when they get it secreted.
Everything in tumor biology goes back to fetal development, and saturated fatty acids are really high for the proliferation that Gang just mentioned. So all these fatty acids are very high at the fetal stage. If we got fetal exosomes from amniotic fluid, I would predict they would be high in saturated fatty acids. And so I'm not really surprised with this result, since the tumor is highly proliferative, similar to the embryo.
Yeah.
Thank you both very much.
Good, thanks for the question, Roger. So Yang Kang, I see that you have a couple of questions, but let's go to, we're gonna go to Phil Askanase first, and then you can ask both of your questions after that. Phil, please go ahead.
Yang and David, this is a very exhausting work. And you've pointed out the target of the tumor EVs and particles is the Kupffer cells. But in some instances, do the Kupffer cells themselves release exosomes and exomeres that cause some of the effects you mentioned, or other effects? In other words, is there an intermediary thing where the Kupffer cells are releasing their vesicles that then cause effects?
Yes, we agree with you, Phil. And there's the paper that came out in Cell Metabolism at the same time. They didn't look at Kupffer cells per se, but they looked at hepatocytes from fatty livers, and those EVs from those liver cells really promoted metastases. So this is something that we want to evaluate and see exactly what you say, how, once the educated liver, especially fatty liver, but we're very interested in how the EVs potentially can change from the Kupffer cell and how they could have further education locally with the hepatocytes, but maybe systemically. So Gang's planning on studying that. Right now we're going to be examining that.
Could you send me the references you've mentioned?
The other paper on fatty liver EVs is in Cell Metabolism, and it just came out in May, June of this year.
Well, do send the references if you can, David. And
sure,
this is a continuation of the fantastic work you've done on EVs and tumors in general.
Thanks for the comments, Phil. Yang Kang, why don't you go ahead and ask both of the questions that you have here now?
All right, thanks so much, Ken. And I have another question. I think the first one for other organs, I think I have the answer. Another one is for the uptake assay, how long for the Kupffer cells to uptake the EVs after your injection? I mean, some paper I saw that they inject for 4 hours and then detect the EVs at the Kupffer cells or some intercellular cells. But I think you did 24 hours. So what's the minimal time for this Kupffer cell to uptake the EVs? When
Yes,
detection.
for the in vivo education, for the in vivo experiment, we always do like the overnight, and the next day we did the flow cytometry. And actually we did in vitro analysis of the EV treatment with the Kupffer cells. It only 4 hours we can see the TNF-alpha expression upregulated. So at least in our hands, the 4 hours we can see the huge change of the TNF-alpha expression. So which means the EV already functionally changed the Kupffer cells.
So yeah. So this is for how about the in vivo? We inject to the animals.
In vivo, we didn't follow up with the time course experiment, but we always followed up our protocol now currently overnight to see their biodistribution.
All right.
I would probably say that the uptake starts in a few hours that quickly, because when we inject, tumor exosomes and we're studying the lung, they can be taken up by endothelial cells and macrophages in the lung even after 1 or 2 hours. So it's pretty quick of the uptake.
Yes, actually, when we do the tumor explant-derived EVPs, like the metastatic lungs, after injection, the mice could be die in like very short time after the injection because of the thrombosis. So I would say, like David said, it's really quick after the EVP injection. So the EVP could be uptake by the macrophages and the tissues.
Yeah, great.
Yeah.
Thanks so much. Good work.
Yeah, thank you.
We also have a question here from Chanan, but I don't think that Chanan is still on the call. So let me just summarize this. Do you think that there's a role for RNA transfer in any of the effects that you see?
Actually, the other postdoc in our lab has done something about the RNA sequencing in the EVPs, and they identified a couple of microRNAs, like microRNA 18. So they are still doing the functional downstream functional study. But for the fatty liver, we didn't check in detail how the RNA can contribute to fatty liver generation.
Okay, thank you.
Yeah.
Rodrigo Kawasaki, could you please ask your question?
Hello, can you hear me?
Hi,
yes,
we can.
Yes, okay. Hello, and thank you for the great talk. And I have a question about the tropism of the exomeres. So you said that they preferentially go to the liver, but I have a question. If you guys consider to analyze the surface content of these exomeres to check if there's like a specific molecule that can target, the Kupffer cells.
Yes, actually, we have done some proteomic analysis of the exomeres, and we did find the potential target protein like the galectin-3 binding protein, which could target the macrophage in the liver. So we are doing the downstream analysis now. We are trying to block the galectin-3 binding protein to see if we can block exomere uptake in the liver.
Yeah, maybe I can make a
yes, yeah.
So besides proteomics, we also characterize the exomere, and together with different subset of exosome, we look at their lipid composition, other modification, for example, glycosylation. So all these can actually contribute to the targeting specificity of these particles. Yeah, that's something we are following up. Thank you.
Thank
Good.
you.
Thanks for that, Haiying. Lothar Dietrich.
Yeah, hi. Thanks a lot for a nice presentation. I'm wondering a bit, you really focused very much on the Kupffer cells in the liver, but you showed in the beginning that liver endothelial cells maybe even take more EVPs up. So I was just wondering, do you think they somehow contribute maybe to the changes in the liver that you observe? Do liver endothelial cells also release TNF, for instance, if they take up EVPs?
Yes, that's a good question. Actually, the endothelial cells uptake of the EVPs, there's some literature that they can also promote the cytokines secretion from endothelial cells. In our hand, we didn't, at least in this study, we didn't focus on too much of the endothelial cells because the other postdoc in our lab focusing on the lung endothelial cells, how it can promote the lung metastasis via promoting the vascular lignins. So we are separate. We are different project in the lab. So yeah, it's a good.
Obviously, they are doing something. We just haven't pursued the endothelial side, but we should definitely look at that.
Yeah.
Right. I'm looking forward to your next publication.
Thank you.
Great suggestion. Thanks. Next is Wei Guo. You have a question about RABs, about RAB27.
Yeah, it's great work. I just have a question about the RAB27. I understand how RAB27 can regulate multivesicular endosome and regulate exosome release. How it regulates exomer release? Can you enlighten us with some mechanism or potential mechanism?
Yeah, this is a really good question because we were also now currently trying to understand how the RAB27A can promote the exomer secretion. And we did the knockout in the melanoma cells, and when we do the F4 like fractionation analysis, we did see exomer is the majority of these particles that are suppressed after RAB27 knockout. So for the mechanism, currently we don't have very detailed, but we predict it might be associated with probably the cytoskeleton microtubes, this kind of thing. So the cytoskeleton might be functional together with the RAB27A to transport the exomer to the extracellular out of the cells. So unfortunately, we don't have the mechanism now. We are still investigating how exomer can be regulated by the RAB27A.
Yeah, I just want to make a point that, we always learn from others that it was the exosome population that changes the most in RAB27. And now we're finding, Haiying could make more comments because she's the one that really defines the fractionator that separates them. But now RAB27 seems to be more related to exomer release and not so much exosomes, especially the larger ones that are 90 to 120. They're not greatly affected. The smaller exosomes are the 60 to 80 size range, but especially the exomers. So does that give us clues in their pathogenesis, which we still don't know too much about? But it kind of tells us that, it's not, RAB27 may not be exosome and larger exosome release mechanisms as we once thought.
So do you know any intracellular localization of exomer? Any compartment?
Haiying, why don't you answer that? Because you...
Yeah, so actually, we can only, so right now we don't have a, detailed answer for that, sure answer for that, but based on our proteomics, we have some hint is from some specific organelle. It may be derived from, for example, mitochondria is one of them. But I mean, this is also limiting by the imaging tool we can use to define exomer, use as a specific marker of exomer and how we distinguish from the intercellular components. So it will be definitely something we want to look after, but it's still very challenging at this stage.
But it will be very exciting if you figure things out.
Yeah, definitely.
Yeah, thank you. Thank you.
Thank you.
Thank you. We have time for about one more question, and we have one here from Alex. Alex, please go ahead.
Great. Thanks so much, Ken. Gang and David, great work. This is really fantastic. So the question that I have relates to differences between mice and humans. In humans, the CYP genes are highly polymorphic, right? So you guys are showing these effects in the liver mediated through the CYP genes. But in mice, we really don't have this issue of different mice having, different variants and therefore different effects. And I'm just wondering, in patients, for example, perhaps this, David, from your work with patients or from your other colleagues and stuff who work with patients at MSK, but do the CYP polymorphisms have an effect on the phenotypes? So, for example, the downstream effects that you're talking about with regard to fatty liver development, cachexia, or some other things?
Yeah, I think right now in patients, we just have the correlation that we described in mice today. So the orthopedic surgeons will tell you in all patients that they operate on osteosarcoma and they only get lung mets, not liver mets. Mm-hmm. They have in children, extreme high fatty liver. So, it's going along with our story, but we just don't have all the mechanisms worked out in the patients yet.
Okay. Yeah. So the sorts of things that to look at, and perhaps at a place like MSK, you guys can do this because you have so many cancer patients, but racial and ethnic composition, right? So for example, do African Americans versus Caucasians versus Asians, are they, more predisposed, more likely to get fatty liver or some of these other effects? That could be something that you can take a look at. So
yeah, that's extremely interesting.
Yeah. Otherwise, thank you so much. Really appreciate it.
Thank you.
Well, still some things to learn then, clearly, and we're coming to the end of our hour here. So we're going to close, but before we do, I would just like to ask our authors, so Gang, Haiying, David, what's a big question that you would like to address that you think is still out there that nobody has really been able to answer about EVs and other extracellular particles? Is there something that you can leave us with to inspire us to get back into the lab and try to answer things?
I just want to highlight there's much more room for the systemic effects of EVs that we haven't even tackled yet. That would be good for especially a young, or I don't believe in age, an old investigator to examine. So for instance, what are these, do exomers or exosomes, what's happening with the brain when this is happening? We do know, Gang has shown, and they commonly go to the adrenal glands, which is a common metastatic organ. But what are they doing to the adrenal glands? Are they supporting all the new work that's been proposed in stress factors that are really increasing metastases? Do they have a role there? So you can ask all these questions. Do they ever go to muscle or not? And why don't they go to muscle? Because muscle's not a common site of metastases. So I'm interested in the whole body, and we really need to figure out, what their contribution is.
Excellent. Well, we will stop there then. Thank you, David, for those comments. And thanks again for the presentation today. Thank you to everybody who joined and for all the great questions. and conversation here. And we will look forward to seeing you at a future EV Club. Thanks again, everyone. Have a great rest of the week. Bye now.
Thank you, Ken. Thank you, everyone. Bye.